Waalaikumsalam warahmatullahi wabarakatuh.
Ini semoga beliaunya nggak lupa ya.
Ya,
Cuma
tadi,
lewat email. Saya cuma lihat
tadi
email
sudah masuk kok Bu tadi Bu.
Oh ya. Oh,
Saya
syukur.
sempat chat sama beliau. Itu ada
Oh,
tuh, kanan paling bawah. Bujang.
Oh, mana? Oh ya udah.
Sudah ada beliau.
Sudah ada.
Ya, sudah ada.
Nanti cuma greeting paling 3 menitan sih Bu, untuk pembukaan sedikit, bukan
Oke.
pembukaan, sambutan sedikit sih, bukan pembukaan, memang kan kuliah.
Punten, suara saya kedengaran jelas nggak ya?
Jelas Bu.
Jelas Bu ya.
Terima kasih, terima kasih.
Alhamdulillah. Ibu atur nuhun Bapak Ibu, Bu Pes juga nggih. Nanti saya join sebentar Bu ya, karena ada meeting lain mau di Cibodas ya.
Ya, monggo.
Oke, paling sebentar nanti habis sambutan terus live.
Baik. Profesor Gujang. Good afternoon, Professor Gujang. Nice to have you here. Hello. Can you hear us? Yeah, we can hear you very clearly.
Okay, thank you.
We are waiting for 1 more minute before we start the meeting, the lecture.
Can we start the agenda?
Yeah, sure.
Okay. Good afternoon, ladies and gentlemen. The webinar is going to start immediately. To all participants, please use the virtual background and fill the attendance form from the link provided by the committee in the chat feature. We will start the session. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon. It is a great pleasure to welcome you to this webinar series ROAFERIAN 3, held by Research Organization for Agriculture and Food, National Research and Innovation Agency BRIN, Friday, September 22nd, 2023. Today we are going to engage a very interesting topic about genome editing on cattle by Professor Bujang from Seoul National University. In this occasion, we are honored to welcome Puji Lestari, M.Si, Ph.D, Head of Research Organization for Agriculture and Food BRIN, Professor Bujang from Seoul National University, South Korea, and our moderator for today, Dr. Insinyur Bestisnamurti, researcher at Research Center for Animal Husbandry BRIN. Ladies and gentlemen, next is a welcome remark from Head of Research Organization for Agriculture and Food BRIN, to Mrs. Puji Lestari, M.Si, Ph.D. Time is yours.
Thank you, Ms. Andalusi. Ya, Assalamualaikum warahmatullahi wabarakatuh. Selamat siang, salam sejahtera bagi kita semua. Good afternoon, everyone, and especially our guest speaker today, Professor Bujang. So first, I would like to thank and deliver my sincere gratitude to you, the honorable invited speaker, Professor Bujang from Seoul National University. I was alumni of that university. It's from South Korea, and also to the moderator, Dr. Bestisnamurti, ladies and gentlemen, who mostly from the Center for Animal Husbandry, I think is under ROAF, BRIN, participating this ROAFERIAN Series 3. So first, let us say grace to the Almighty God, Allah Subhanahu wa Ta'ala, that as His will that we are gathering today to take, to make happen this event, this guest lecture with the topic of genomic editing on cattle by Professor Bujang, Seoul National University. So please just allow me to explain briefly about this GA. So this recent development of genome editing systems dramatically changed the field of genetic engineering. So the technology can increase the efficiency of introducing targeted modification in livestock and have significantly reduced the time required to produce genome editing livestock. That should be expanded their use, including in agriculture. So the application of genome editing in the tropics, including in Indonesia, is not only for animal breeding. These tropical pastures can be also edited to increase drought tolerance and the stability to provide good quality forage to feed animals throughout the year and their health. And therefore, as with earlier genetic engineering process, whether breeders will be able to employ genome editing in cattle, genetic improvement programs will very much depend upon global decision around the regulatory framework and governance of genome editing for food animal. So Seoul National University has long collaborated with several universities in a research institution in Indonesia. At present, the National Research and Innovation Agency BRIN, Republic of Indonesia, as the only governmental research agency in Indonesia, is an open platform and is highly recommended to international collaboration and strengthen network, including the Research Organization for Agriculture and Food ROAF. So this way, today ROAF intend to consistently improve the knowledge of researchers for cutting-edge technology. This is inside of the topic of genome editing on cattle by Professor Bujang. Thank you again for your availability and could be beneficial for breed researchers to increase cattle productivity in tropical environments in Indonesia. So ladies and gentlemen, therefore today's lecture is expected to provide participants with alternative perspective, insight, and personal experience of the lecturer, Professor Bujang. And second, to open opportunities to internship and capacity building. Also, we would like to strengthen networking and initiate collaboration and cooperation research with Seoul National University. Finally, I wish you all a most fruitful and enjoyable lecture. Thank you in advance. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Mrs. Puji, for this speech. And to all participants, before we start the discussion, we will have a photo session first. Please activate your camera and for the committee to take the screenshot. I will start count from first slide. Ready? 1, 2, 3. Second slide. 1, 2, 3. Third slide. 1, 2, 3. Thank you. Ladies and gentlemen, now we are going to start our discussion for today, which will be led by Dr. Insinyur Bestisnamurti. Before that, I will read her CV first. Dr. Insinyur Bestisnamurti, Master of Science. She is a researcher from Research Center for Animal Husbandry, Research Organization for Food and Agriculture, BRIN. Her area of expertise is exploration characteristics of local animal genetic resources. She got bachelor degree in animal science and fishery at Diponegoro University, master degree in animal science at University of California, Davis, and Ph.D. at IPB University. Her career track are Head of Program and Evaluation of the Indonesian Center for Animal Research and Development in 2007 until 2010, Director of Indonesian Center for Animal Research and Development in 2010 until 2016, Research Center for Agri-Industry, Research Organization for Food and Agriculture, National Research and Innovation Agency, BRIN, from 2022 until now. She already published 112 scientific papers and contributing author in several books. So without any further ado, to Mrs. Bestisnamurti, time is yours.
Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Assalamualaikum warahmatullahi wabarakatuh.
Yeah, my name is Bestisnamurti, and you can call me Bes, and I will your chairperson for this afternoon lecture. Welcome to Professor Gujang, DVM PhD. He's a lecturer from the Department of Theriogenology, College of Veterinary Medicine, Seoul National University, Korea. And this is very excellent opportunity for all of us. The National Research and Innovation Agency, Republic of Indonesia. And we also have other participants from different research organizations. Today, Professor Gujang will present his lecture entitled Generation and Germline Transmission of Genome Edited Cattle. Let me read very briefly his CV. He had his Ph.D. degree in 2006. Okay, thank you. Okay, already published like 257 international scientific papers and contributing authors in several books. And he also has experience in generation of double knockout cattle via the CRISPR-Cas9 RNP electroporation. There are many others of his publication. I try to count his publication from the CV. It's about 75 paper. And his area of research skill is mainly for germ cell manipulation, general surgery in theriogenology, embryo transfer and its relevant techniques, basic molecular works, also the stem cell. He also have like 9 awards and scholarship around the world. And he also already produced like 5 patents. So without further ado, I would like to give opportunity to Professor Gujang to have your presentation. Can we have like 60 or 75 minutes lecture, and then we will continue with the discussion. And then for all the participants, maybe we wait until all the comment and question until at the end of his lecture.
Ada yang lain?
It's time for you,
Pakai apa?
Professor,
Bisa.
your lecture. Ibu minta tolong di off-kan lainnya ya Bu, excuse me. Terima kasih. Thank you, Professor Gujang. No time is yours.
Thank you. Good introduction. I'm happy to sharing the progress in genome editing in cattle. Now I am in Australia, not Korea. I was speaking about my recent result in the Pacific plant and genomics. Maybe this is the conference. I was here, Australia, Perth. Yesterday, I was sharing my result about that. Again, thank you for inviting the speaker in your BRIN. I believe the BRIN is the most pronounced the institute in Indonesia. I have the good friend in Yogyakarta, Agung. I don't know full name, Agung, Professor Agung.
Yeah.
He's my friend. We was together in Japan 10 years, around 10 years ago. Yeah.
Yep.
Still, I am communicated with him. And also another Professor Yuda Fabrianto in Yogyakarta.
Yeah.
So also I already mentioned about right now I am a adjunct professor in the Erlangen University.
Yeah. Last year you were there, yeah.
Yeah, I was there. I'm trying to do November. I have the plan to visit Indonesia to make some project, because in Korea government and very big pharmacology company, we wanna developing some co-project with Indonesia. So maybe I'm gonna sharing the project profile 2 weeks later to BRIN. Anyone interested about the genome engineering in stem cell or embryo animal, you can apply the pharmacological project. It will be great chance to collaboration to future, because this is a good chance to collaboration, because we provided a lot of money to the research. Probably I'm going to planning to visiting the Bandung Institute of Bandung. I don't know exactly. Yeah.
ITB.
I will go there firstly, and then I'm gonna plan to several. We gonna right now my secretary screening which university or which institute will be best partner to produce to project our genome engineering one. I will sharing in the future.
Okay.
So today I just show some my genome editing study today. Maybe I will sharing the screen. This is the
screen. Yeah.
Yeah. Okay. So anyone interesting about probably many people, everyone will be interested in genome editing, because the 2012 came out in the mammal cell. So right now a lot of country this genome editing apply to plant, animal, and human medicine, or the other some diagnostic tools.
Yes.
So in my case, I was starting the genome editing from 10 years ago with the senior scientist in my university. And then he also interesting about genome editing application to livestock. So we was collaborating still to produce the genome edited livestock. And firstly, I will successfully many genome edited cattle was born, and that edited cattle are grown up to date with the normal grown up and normal healthcare, and their germ cell means that we collecting the semen and oocyte, we find out the mutation was proved that. So we believe the genome editing will be very quickly spreading the whole of the world, because the artificial insemination can be possible. We already some genome edited semen was frozen in liquid nitrogen.
Excuse me, can you move the slides, please?
I will slide.
This is
So,
still the first slide.
yeah, I'm just
Okay,
briefly.
thank you,
Yeah.
thank you.
My university, maybe when you can easily find out my university. It's Seoul National University. Is located in the main capital city. So this is the main gate, our university. Maybe some of them have any, if there will be any experience to visit our university, you can see the main entrance, this one. Another one, our veterinary college of veterinary medicine, our school, only one American Veterinary Medical Association accreditation university. Because it means after graduation, our Seoul National University Veterinary School, and then every student to apply to the certification to American veterinarian. So still, right now, our university fully focusing to developing the veterinary education, how to improving the future generation. Meaning we very interesting how to educate the next generation. So let's move more practically to focusing the genome editing in cattle. Everybody will know that some cattle will be one of the most important animal, because they will be provided the milk, meat, leather, a lot of things to human beings. So long time ago, around the 10,000 years ago, I don't know exactly, but the literature review, they said the cattle, the wild cattle, was domesticated around 10,000 years ago. And then the human is starting the artificial selection based on the phenotype. And then we are trying to the crossbreeding, and let's start some growth record, which phenotype will be grown up good with the good trait. After that, a lot of kind of the assisted reproductive technology. The main one is the semen bridging. So the semen bridging means that we can do artificial insemination all of the cattle. So this artificial insemination now is the widely spread in the general farm. I don't know how much percentage this artificial insemination working in Indonesia. In case the Korea, over than 95%, almost every the farm, cattle farm, they employed the artificial insemination. Nowadays, some of them in vitro fertilization, multipopulation, embryo transfer, even the sexing technology. Another one, the dolly, cloned sheep, the nuclear transfer is also developing some reproductive technology. This is the correlation of the induced pluripotent stem cell. This is can be nowadays the therapeutic area. And then the research tools like the quantitative trait, marker assisted selection, genome selection, a lot of the omics data, transcriptome, proteomics, epigenomics, metabolic, lipidomics. I don't know, but there are a lot of omics data came out. So both of the tools contribute to the breeding with the high genetic trait. And then finally, very recent came out the tools we called the genome editing. The representative tools, the zinc finger nuclease, TALEN, and CRISPR-Cas9. So those the genome editing, those two tools will be contribute to the designed breeding. Somebody called the precision breeding. So we can do some specific trait in cattle. So I briefly reviewed some general, the historical, the breeding program in cattle. Everybody well known though in the future, it was starting the long time ago. When I was young, my mom and father, we was raised some cattle. My fathers and mothers, they did the natural breeding, like based on the phenotype. So the female, male, just select and breeding. This the natural breeding still some country or some underdeveloped country, also even Korea, very limited farm, they applied this natural breeding because this is easy, low cost. The pros are not this, but the cons very ineffective program because the single pregnancy, long gestation, long-term monitoring the phenotype. And then another breeding program, well known, the artificial insemination. So after came out, the semen freezing tools came out, and then scientists frozen the semen and then shipping all over the world with the high performance trait. Probably the Indonesia, I believe some of the semen was imported from the USA or Australia, other country for improving the genomic trait. So the frozen semen was inseminated the female, and then we generated a lot of the calf, and then selected some high performance female, and then we again another semen was inseminated. This product program have the pros and cons. Pros is easy, we can quickly inseminate it, but cons is the male dependent breeding. Usually we just considering which male will be best for female. So right now, the AI program, artificial insemination program, most of the farm in all over the world will be applied today. Another some challenging about the time-fixed artificial insemination. Look at this slide. This is the estrus detection. Still many farm, they did the estrus detection based artificial insemination. It means owner of the farm find out the hitting, estrus hitting, and then call to the veterinarian or technician to insemination. But nowadays, this is very labor, and it needed a lot of the cost effect. So developing the time fixed the artificial insemination. It was grown up. Maybe you can find out the more literature review about the time fixed artificial insemination. It's a science will be going on. So another tools like the multiple ovulation, we call it the ovulation, and embryo transfer breeding program. This is the unique program because we can selecting the female and male. Previously, I mentioned about the artificial insemination. It's very easy, very good program, but the disadvantage, they have the only male dependent breeding. But this is in case this one, we can selecting the female and male together. So special program to cow induce the multiple ovulation. There are one of the representative the hormone program. So you can find out a lot of the super ovulation program in the website and injecting the hormone. For example, the progesterone releasing device implanted the vagina and then remove the pre-cycle and then injecting the estrogen and GnRH. And then we're waiting some multiple ovulation. And specific the male one inseminated and collecting the in vivo embryo in uterus. This the embryo transfer the recipient. So this in vivo embryo transfer very impact for improving the genetic trait in cattle until the came out the OPU based study. The next slide I'll mention that. This the in vivo program has the pros and cons. Pros is the high pregnancy, improving the male and female trait. Cons is about one of the issue, sophisticated skill embryo collection. I believe that any experience with the embryo transfer and collection with the like this the superpopulation is very difficult. And another one is the cost issue. We have to inject the hormone. It will be correlation about the cost issue. And listen to some technological advance with the technical advance. Most of the scientists nowadays in focusing the in vitro embryo production system. We call it the ovum pickup system. It means we collecting the oocyte from the slaughterhouse ovary and then live animal based on the ultrasonography guided. So immature oocyte was collected the female one, and this immature oocyte was incubated the in vitro laboratory, and the frozen sold semen was fertilized in vitro. We call the in vitro fertilized embryo. There will be embryo grown up in vitro culture, and then selected the high quality embryo transfer the recipient. This is the powerful tool nowadays because this is the listen to the high pregnancy rate and consistent embryonic development contribute to the improving genomic trait. The cons, the low pregnancy 2010, but right now is the high pregnancy is changed like that. And another cons is the higher skill for require the in vitro production and culture. This one. So anyone interested about how to learn the OPU and in vitro fertilized, please contact with me. We can educate the program in my laboratory. So this is a very good information to you. This graph was show the in vivo embryo production. I already mentioned about in vivo embryo. This one is the collecting the data was the International Embryo Transfer Society rules and error. So times going on, the in vivo embryo production number, it was decreased. However, OPU based, it means the in vitro fertilized embryo based production number is very quickly the increase. So 2017, at that time, the OPU based, in vitro based embryo production number is much higher than in vivo embryo. So this is the total number of the embryo production in vitro. So gradually the increase, the time going on. So probably 2012, we can see more number of the in vitro production and in vivo production embryo. So assisted reproductive technology have a great tool in the livestock improvement compared to the other livestock. Cattle has a long gestation interval, low fecundity, is a slow genetic progress. So we need a new strategy improving genomic trait. So this is the current program. I already mentioned about that. So this is the current donor selection. We have to selecting the female and male. And in vivo embryo production, in vitro embryo production. Finally, the blastocyst embryo was the collect and cultured in vitro. The selected blastocyst and transfer the recipient. And after that, pregnant cow will be delivered the elite calf and grown up and phenotype and genotype based selecting the cow and bull, and then again donor selection. So here I will be combined some genetic tools. It means that we can getting the some molecular biology and biotechnology tool. For example, pre-implantation genetic tools and freezing. Some of the blastocyst was frozen the in vitro embryo, and then pre-genetic diagnostic to find out the sexing or genetic digit in embryo. Another one, the primary culture to skin these elite couple and clone, and we can make some female and male one. Another one, genetic engineering tools like the transposon, virus, or nanoparticle. A lot of the genome tools, including the genome editing one. So I will do what I am doing in the future. In long times ago, my laboratory starting the 20 years ago, focusing the bovine embryo. Firstly, initially we was focusing the multiple embryo transfer. It means the in vivo embryo we did. Another one, in vitro embryo production. We're focusing the some culture media. So my colleague, studying the bovine embryo, in vitro embryo gene expression profile with the different culture media. Finally, we have some special media to show the high development competence in vitro. Look at this video. We used our culture media support the embryo to be blastocyst with high efficiency. This cultured media without any FBS, any serum, that will be great because the no serum means the cheap price and chemically defined one. And then this is the freezing program. After sewing with 24 hours, during 24 hours, the embryo was captured by live imaging camera. And then look at this, survived the embryo. Over than 90%, most of the embryo survived after sewing the freezing embryo. Based on those kind of in vitro embryo production technology, I'm moving to the transgenic technology tools. So my excellent former Ph.D. student, Suyang Yam, she was the background. She has the scientific, the molecular biologist. She was joined my laboratory. We're gonna apply the transposon-based genome tools. This is transposon-based genome tools, and then we generating the over than 9 number of the transgenic cattle at the time. And the transgenic cattle was grown up, and then we follow the long-term health and germline transmission. So that will be published 2018. So at the paper, we was follow the 2 transgenic cattle, female and male, and natural breeding, and generating the next generation. We called the SNU F1. This cap was collecting the blood. The blood from the genomic DNA was extracted the blood, and then the genome was analyzed with the next generation sequencing. We call the NGS sequencing. That sequencing data show the genome stability. We didn't find out any significant difference between the wild type and transgenic one. Finally, we collecting the semen from this F1 because F1 is the male, and the semen was frozen dead. So we did the in vitro embryo culture. So look at this one, wild type semen and GFP semen. We look at this, the great GFP expression in vitro blastocyst. So nowadays, we use only IVF and selecting the GFP plus 1 and transfer the recipient. We can, and then that selected the GFP embryo will be transferred. And then next, another my student, PhD student Kyungmin Kim, he was the finalized the F2 generation bovine embryo with the stable germline transmission and maintenance of transgene expression through the 2 generation. I already mentioned of the founder animal, female and male, generating natural breeding, F1, and then we're collecting the semen, and then wild type fertilized. We find out the conditional lock embryonic expression in blastocyst. That will be embryo was frozen, and the semen was fertilized the wild type and transferred the recipient. Finally, we got another one, F1 generation. The collecting the fibroblast was frozen here. So we have the resource to study in vitro, like this, the green fluorescence expression, semen, and the fibroblast. After that, after completely research done transgenic animal, and our laboratory is focusing to genome editing. So Kyungmin Kim, he is my PhD student, already graduation. He got the degree. He is the grown up in the farm because his father operating is a farm. So his childhood was played with the farm and a lot of the cattle. So he is very interesting about the genome editing one. So he did it in vitro fertilized embryo and injecting the microRNA to deleting some gene. It means that we call the genome editing. So for initial stage, we candidate gene with MSTN, myostatin. This is very famous gene because the myostatin is the negative the muscle inhibitor. So if the myostatin will be mutated, it means the muscle growing will be muscle will be highly grown up compared to wild type. So we designing some Cas9 and guide RNA to targeting exon 2, injecting the embryo and collecting the blastocyst. Some selected blastocyst to transfer the recipient. And then finally, we got some MSTN knockout cap. Look at this, the imaging shows this cap. The picture was captured 2 months old. The outgrowing muscle is easily detecting a lot of places. This picture is captured 25 days old. Still, we can see outgrowing muscle all over area. Somebody wondering the myostatin knockout have some health problem. So regularly, we visiting the farm to monitoring the health issue by analyzing the blood analysis. So this is the general general blood analysis. Look at this one, WBC, RBC, and a lot of the serum chemistry. So there will be any significant difference between the wild type and knockout. And then the female and male knockout, the cattle is grown up over than 1 year. It means they will become a puberty, and we starting the next generation. So from the female, the ovum pickup was carried out to collecting the immature oocyte, and from F0 male, we collect the semen and in vitro fertilized. We analyzed the knockout F1 generation blastocyst. Look at this, the data. The blastocyst well grown up. and mutation, we find out and transfer the one embryo, and we find the one pregnancy was found in this paper. At that time, we find some problem with the microinjection, because the microinjection, it needed the expensive micro manipulator, and another one very difficult to handling RNA. So our laboratory, the excellent student, suggested the electroporation with the Cas9 protein and guide RNA. It means the general electroporation. Electroporation means the very simple tool to delivering the guide RNA. So we can easily mutate the double knockout. So we did the embryo transfer more here. We did it at the time. We just showed the pregnancy, but after that, we collecting the more oocyte and fertilize the embryo and transfer the recipient. We find out the 3 F1 generation, the two female, 1 male. They be grown up 1 years old. Probably next month we're gonna do collecting the oocyte, and future will be collecting the semen for the next generation, F2 generation. I didn't publish about that. We did whole genome sequencing from the F0, F1, the sequencing data. We didn't find out any genome problem. There Cas9 effect. Another one also, we collecting the very tiny tissue, muscle tissue, and look at this, the muscle bundle, the knockout, the cattle muscle bundle. We can see number of the muscle bundle increases that, and muscle fiber is the thinner compared to the wild type, and myostatin expression level decrease. Another very interesting one, we collecting the stem cell, the muscle stem cell. This is from the knockout. This is the wild type. So we didn't find out a difference, and time's going on, and after almost day 6, we can see well differentiated muscle fiber in knockout one. It means our knockout, the muscle myoblast, can be easily differentiated into myocyte compared to wild type. Another, we keep going project about proteomics to compare to the difference between the knockout and wild type. That data will be came out end of this year. My colleague, he's doing analyzing the proteome, all proteome in muscle between the knockout and wild type. And another project was parallelly our laboratory was did. Some excellent student, Kyung-hyun Kim, was joined together. We was here, did the paper. We was applied to a lot of the high efficiency knockout one. At that time, we did the double knockout strategy. One strategy about beef cattle, the other strategy is the dairy cattle. For beef cattle, 2 candidate genes, myostatin and prion, will be electroporated the Cas9 protein and RNA. We find out high efficiency single and double knockout, and those embryo was transferred to recipient. Finally, we got it. The same strategy was applied to the dairy cattle, different gene, myostatin and beta-lactoglobulin. Beta-lactoglobulin, one of the major allergen in milk. Some of the people don't drink milk because this allergenicity. So we believe that the beta-lactoglobulin knockout the cattle, good choice for future milk consumption. This is the summary data table about the double knockout study. and the knockout mutation efficiency. And this is the beef cattle and dairy cattle. And the two male beef, dairy, both the male grown up, right now 12 years, 1.2 years old. And then we're collecting the semen. We're collecting the semen and the frozen in liquid nitrogen. After sowing, the sowed semen was fertilized in wild type site, and we check how difference wild type and the double knockout semen activity. So we find out the double knockout in semen, myostatin, prion, and myostatin beta-lactoglobulin, double mutation. Another one, we keep going to monitoring the healthcare regularly, usually 3 or 4 months. The clinical team visited the farm to collect the blood, to analyzing the blood chemistry. Currently, there is no any problem in blood analysis. It means that they are grown up normally without any issue. This is the summary of the engineered cattle. I mentioned firstly of GFP, reporter cell and semen, myostatin Cas9 based myostatin knockout cell and semen, and myostatin and prion, double knockout cell and semen, myostatin beta-lactoglobulin cell and semen was frozen in vitro. So I can sharing for future collaboration. And I didn't show this data because recently this year, beta-lactoglobulin GGTA1 knockout cattle was born. We collected the semen and analyzing the double mutation. And finally, we got it. And another double knockout, GGTA1, CMAH, both of the candidate gene for transplantation issue and or allergen in meat allergen. We successfully the double knockout was generated by RMP protocol. Another one, Cas9 expression cattle, we collected the cell and semen. Lastly, I was showing some two big project, ongoing project. One is the long-term monitoring. Our first transgenic cattle at that time that was published in 2016. That I mentioned about over the 9 transgenic cattle was born. So F0 first was born. She is now 10 years old without any health issue. Another male one was grown up until now. He is 9 years old and still grown up with the normality. And then there will be lateral breeding. The F1 generation was born and he is 7 years old. So we called the transgenic family. This the family, mother and father and son. This family will be happily well grown up currently. I will do next year, publish the very simple report, what change in 10 years after 10-year transgenic cattle. Another one, we did some all-in-one Cas9 protein. So Cas9 will be injecting the embryo and generating the F1 generation and collecting the semen and in vitro fertilized, generating the F1 cattle and grown up and selecting the F2, generated the semen and frozen the liquid nitrogen. Another one, we want generating the all-in-one knockout plasmid for generating prion knockout cattle to study mad cow disease. We called it the BSE, bovine spongiform encephalopathy. So successfully, we generating the knockout prion cattle. He is now over than 2 years old. We collecting the semen from the male one. And we transfer the embryo, and then F1 generation male calf was born. We're collecting the semen and fertilize the embryo, and we can see GFP expression very well. It means the GFP and Cas9 was the co-integrated this urinum plasmid. So GFP expression means the Cas9 was the co-expressed. So we just the guide RNA injected the embryo, we can easily the knockout. This is a study about the data. And final the slide. I was developing some designed breeding center, another called Precision Breeding Center. This is located in the middle of the Korea, South Korea. We now starting some sequencing to superior phenotype, inferior phenotype to find how big difference between the superior and inferior individuals. And also we're gonna do compare to the genome editing derived phenotype. So how difference between the two genomic studies. So and then we're gonna do collecting the oocyte and fertilized by ultrasonograph in here Precision Breeding Center and transfer the recipient. This is our big project to keep going on to future designed the cattle production. Thank you for your attention. So I will be very thank you to the university colleague, the co-founding research company, and Gyeongsang province the scientists. All of them, they will be supporting our genome editing progress. Thank you for your attention. I will be happy to take a question. Anyone?
All right. Thank you so much, Professor Gujang. Very interesting research that you have been produced from your laboratory. It's now time for comments and maybe question. So I will just give opportunity to our colleagues to raise comments maybe. Bapak Ibu sekalian, silahkan untuk menyampaikan. Can I speak in bahasa Indonesia, Profesor Gujang? Bapak Ibu sekalian, silahkan untuk menyampaikan komentar dan pertanyaannya. Bisa ditulis di chat box ataupun langsung untuk raise hand. We have our director, Dr. Puji, in our webinar, so maybe he would like to start with some comments and question. So, Pak Puji, time is yours.
Baik Ibu Bes, thank you Dr. Gojang. This research is very interesting. We at BRIN are just starting to do research into the genome editing, but we still have to improve our researcher capabilities. I hopefully our researcher can join to your university, for example, for a postdoc visiting in there, because for cattle genome editing we have no program for this research, but for plant genome editing is have running to research for this program. It's possible to our researcher apply to postdoctoral in your university.
Yes, welcome. Welcome. Any interest about the bovine embryology? Because I was the whole life was the correlation with the bovine embryology.
Any other question Pak Puji would like to ask to Dr. Gujang? You're still
cukup Ibu Bes, ya, saya pikir cukup.
Okay, all right. Professor Gujang, I look at your presentation, like the slide number 3, you mentioned about how in Indonesia artificial selection or breeding assisted reproduction technologies that's already there. So maybe I would like to just give a comment that Indonesia have like produced almost 4 million frozen semen distributed to the whole country. And you are right that most of our bulls come from Australia, but besides that, we also have local bulls to be the donor for the semen. Yeah, so the total frozen semens we have only 4.5 million approximately. And we expect to have like 80 to maybe 85% of success rate to have the calf, every year. And about the embryo transfer, we have one institute we call it like Institute for Embryo Transfer. It is under the Ministry of Agriculture, under the DG of Livestock and Animal Health. So they have different kinds of donor females from different breeds. They also have like dairy, local breeds of cattle, and they have like the target production of embryo, something like 500, 250, or something like that, the total numbers. They also have the target to have like transfer embryo in several farmers. And I think maybe the target for the transfer embryo to be calves, something like 200 calves born in every year. This is very limited number actually, but this is about I can tell you. The IVF may be only in term for the research. Yeah, we did it in many university. We also have like sexing studies, but I don't know exactly how many of them to be into production. Okay, we have like one participant raise her hand. It is like Ibu Pradita Lustitia. So time is yours, Ibu. You can just open up your camera and unmute it. Okay, time is yours, Ibu.
Okay, thank you, Bubes. Good afternoon, Dr. Kujang. For introduction, my name is Pradita Yustisa Tarsmi. I'm the junior researcher in ORPP, especially in Animal Research Center. My question is simple. It is because there is genomic editing, there are any potential effects that occur or are being researched in your group about the reproductive parameters such as reproductive organ or hormones in the result in the offspring? Because some studies have stated that genomic change could have an impact on the reproductive ability of the offspring or the calf. Because I'm from the reproductive group, I just have those questions.
Okay, good point.
Okay.
So you mentioned about the genome editing in the cattle. You are curious, is there any problem with the reproduction?
Yeah.
So currently we was generating the 6 kind of the knockout cattle. It means the genome editing 6 gene was the each species, each gene was knockouted there. But in vitro level, it means that usually the in vitro fertilizers was there. So we didn't find out any fertilized activity. So semen, mobility, and semen, a lot of the parameter was analyzing by computer system. There will be any problem to compare to other species, other wild type one. And in oocyte, so we didn't compare to the oocyte, but after fertilized in vitro embryo development, there will be any significant difference. So we believe that there will be no problem. But in kind of the academical approach in kind of the steroid hormone, like the estrogen level, progesterone level, testosterone level, and endometrial hyperplasia, more ovary changing. We didn't analyze that, but in the future, because we have the only limited number of the knockout cattle, particularly the female, so we just regularly capture the ovary, to ultrasonography. What's changed in ovary means the follicular growing and ovulation and growing of the corpus luteum. So currently, very limited study, there will be any problem in reproductive competence. But we hope we future our one of the project is the long-term monitoring over than 10 years. What's the impact on the reproduction? But right now, everything fine.
Thank you, Dr. Kuchang.
Okay. Any other, Ibu? Okay. Since we are waiting for other participant to raise comment. Oh, there's one participant. Pak Suyatno, you want to raise a question? Please, time is yours.
Okay, thank you, Dr. Best, and thank you, Prof. Kuchang, for the nice lecture. Yeah, I have some question about your lecture. Actually, I want to ask about your suggestion for us in BRIN that we start now, just starting the it is like a long dream to have a genome edited cattle. So at first, I will ask. I have two questions for you. First question is about what is your advice for us that we have not just starting for the some in the near future we have actually we hope we will have the genome edited cattle, but for now we just start from the collecting. We have some project that just collecting the stem cell, including spermatogenetic stem cells, also mesenchymal stem cell, and for us, it's better to start. Which point we have to start? And from your lecture, if I'm not mistaken, I can understand that at first you establish the GFP express cattle, and then you use the offspring of this cattle for the genome edited cattle. And what kind of method that we can use starting for genome edited cattle, edit in the embryo, or we can use the stem cell for the edited genome editing? And this is the first question. Hopefully you can catch my question. And the second is, you mentioned about many successful of the genetically engineered cattle in your experiment. So what is the success rate of the transgenes integration in your genetically engineered cattle in your experiment? Thank you for time. That's my question. Thank you, Bubes and Prof. Kuchang.
Okay.
Okay. Because sharing some experience about the genome editing, because that genome editing project in my laboratory was going back to almost 12 years ago. I was starting 12 years ago. So initial stage, my student and technician, we did the zinc nuclease, Thailand, CRISPR-Cas9, but at the time it's very difficult to managing get the high efficiency in bovine, because there will be no any cell line. In human or mouse, there will be a lot of the kind of the cell line, like the HeLa cell, the cancer cell. So there will be cell line is the much easily handling, and the cancer cell show the high transcription efficiency. It means the much more easily generating the knockout one. However, in primary cell, like the bovine, it's an embryo, even though we called the bovine embryo, it is also primary cell. So in case of the primary cell, it's very difficult to managing to introduce the transgene or any genome editing tool. So maybe you're gonna do starting point firstly to optimize the condition. It means the which gene. Firstly, you can starting the some like us the GFP every time visiting our laboratory. Anyone I'm teaching the student and technician. You are starting the transduction with the GFP. So GFP transfection after cell selection and screening over than 60%, for example. Oh, you can now is the ready to genome editing into primary cell. And there will be also another practice about the in vitro embryology. Our laboratory every week, 2 times, we did the in vitro fertilized embryo to test the general embryo production, and another one, the knockout efficiency. So knockout will be very frequently was evented after fertilized, mostly S page, you know, the cell cycle, M page, S page like that. S page highly frequently mutated there. So every laboratory, they have the different protocol for artificial insemination, in vitro production. So after fertilized and monitoring what time the S page entering. In my laboratory, usually after inseminated in vitro, there will be 15 or 22 hours between like that. We did the genome editing in there. Yeah, like this, even every step was optimized per the laboratory specific. So I don't know your institute how to practice the genome editing in bovine cell, which cell. So stem cell is not good for the practice. So most good cell resource from skin fibroblast. Skin fibroblast is very easily grown up, and they will be grown good resource to test genome transfection and genome editing efficiency. That will be first question answer to you. Second, what is the second one?
The second is about the success rate of the transgene integration in your experiment actually to generate the genetically engineered.
Yes. The second successful story is that we have a lot of high efficiency, but the initial somebody student or very beginner came to the laboratory. He or she just showed the 10% in blastocyst, but after 1 year's practice and the training, there will be showed over than 60 or 70 mutation in blastocyst age. And we collaborating some clinical team. We have the very well training the embryo transfer member. He did our knockout embryo and transfer the recipient. He showed the initial pregnancy rate 60%, 60%. So that's why I can generating the many number of the genetically engineered the cattle. So you are also interesting about the genome editing one. You have to making some very well training the embryo transfer team. Particularly selecting the recipient. One of the good point to pregnancy the rate. So I don't know your embryo transfer team how to do embryo transfer in the recipient. Usually in my research team, we was synchronized. We initial stage with the 20 recipient was selected and our synchronized program. And then finally, we find out around the 70% recipient was synchronized. And then we was evaluated corpus luteum,
Mhm.
that cow will be
Populated.
will be good or not for recipient. And then usually we only 60% recipient was used for embryo transfer. So very selective the recipient show the high pregnancy rate. So two team, one in vitro embryo team, the other one embryo transfer team. Two team well coordinated, and that will be show the good result with the high speed and high efficiency. That will be okay to answer.
Yeah. Pak Suyatno, you are still muted. Can you unmute first, Pak Suyatno? Yeah.
Okay, thank you, Professor. I'm just waiting for part 3 for next collaboration. If we can collaborate for establish genetic engineering cattle for Indonesian indigenous cattle. So we probably, if you visit Indonesia, we'll meet. About part 3 is our head of institute, so probably you can meet him and discuss about the next collaboration. For us, probably teach how to do that. So,
Yeah, plus
Okay.
initially you can come to the lab, because look at the eye, just look around our facility, how to do that will be much helpful to your study, because I'm explanation to information to you, but this is kind of just a text, not real. So if you have any chance, you can visiting our lab, or also I can bring my some core member team visiting the institute to helping how to improving, how to optimize that. Maybe there will be good advance for future. Like the online communication, there will be just the communication is okay, but real practical advance, just the visiting is it must be necessary.
Yeah. Okay. Thank you. From the second comment from Pak Suyatno. Just before we move to the next one, Pak Kahim, maybe I would like to keep in mind, Professor Gujang, that seems to me our research institute would like to have collaboration with your institute. That's why in your next visit to Indonesia in November this year, maybe we need to have set your time to see us in Bogor, which is like not far from Jakarta. So maybe Dr. Putri Puji will be in touch with you to set your time and to see us. If that's possible, so we will just continue. Is that okay,
Yes,
Professor Gujang?
it's okay, but my time is very limited. But if any chance to time, maybe we can see.
Yeah.
I also want to visiting your institute. I don't know the BRIN, where it is BRIN.
Yeah, this is like new government research agency just started like 2 years ago. So it just like accumulation from different research agency from different ministry. Okay, let's now move to Dr. Fahim Khan. If you have any comment or question to Professor Gujang, so time is yours. You can unmute it first. Okay, go ahead.
Thank you very much. Thank you very much for giving me this opportunity. It was a really interesting talk by Professor. I have a couple of questions here. So the first one is about the CRISPR-Cas9, editing. We do know that it do have some off-target effects. So I just have question that whether you have some specific considerations to control the off-target effects. And second question relates to microbiomes. So did you have a chance to study the microbiomes of the genome-edited cattle?
Sorry about that. Yeah. First question, to do the CRISPR-Cas9, you're gonna designing the guide RNA. You have to selecting the good guide RNA. Good guide RNA means the low level of the off target. So we generating the over than right now 20 over than 20 mutated the cattle, 6 kind of the gene. So only one cow showed the 1% off target effect. But I don't know. We will be keep monitoring that off target will be impact on next generation, because the very recently one calf was born with the very low level of the off target issue. So but the other one, we didn't find one. So your first question, to answer my question, first the designing the guide RNA, and selecting the guide RNA with the low level of the Cas9 off-target effect. There are several kind of the website to synthesize the guide RNA. So you can use the some good tool to choose the guide RNA. Second question is about microbiome. I want to do that, but I didn't have any microbiologist in my surrounded by me. So if any chance to sharing the lumen or urine or feces, the collecting the microbiome, how big difference between the wild type and knockout, it will be also
like
one of the interesting one.
Yeah, like Patri Puji have this whole team working here with the microbiomes, so we can have this collaboration.
Yeah, you can come. I don't know how to collecting the microbiome. You can come to the laboratory and collecting the microbiome and bring your laboratory analyzing that, and showing the how big difference, how difference there.
Yeah. Thank you very much.
Thank you.
All right. Thank you, Fahim. Any other comment, question from other participants, maybe? Yeah.
Pandang.
Yeah. Oh, okay, Pandang. Pandang. Okay, Pandang, go ahead, Pandang, if you want to raise question. Okay. I'm sorry, I didn't see your.
Okay, it's okay. Thank you, Ibu Bes. I am really appreciate about this lecture, Prof. Kujang. I think this very interesting topic for us. As we know, we are just studying for the adaptation. So as we know, in Indonesia, the transferred embryo came from the outside. I think from subtropical, for example, from Australia. And maybe the problem is the adaptation, the cattle, if you grow in Indonesia. So would you have the experience about the adaptation, about the genetic for specific genetic for adaptation of the stress? I think especially for the stress in cattle. I think it's very good for us, the information. Thank you, Ibu Bes. Thank you.
Okay. Yeah, go ahead. Thank you.
Yeah, you mentioned about the embryo transport. In South Korea also, we are imported some other breed. Recently, Georgie, the Holstein, Georgie, Brown Swiss, different kind of the dairy breed. So maybe Indonesia also, Holstein is the over than most dominant breed in dairy, in even the Korea. But in Korea, we was trying to give the genetic diversity. So we imported the embryo from the Canada and the Australia, the different dairy breed, Georgie. Initially, the frozen embryo was the shipping by airplane, and then arrived to the Korea, and after sewing and transfer the recipient long time ago. This is the long time ago story. At the time, only 10% successful rate.
Mhm.
So people is wondering why the frozen and sold embryo efficiency is very low. So at the time in Korean, the veterinarian and related to some recipient prepare technician, there will be managing some recipient synchronization program, selected program. Then right now, the same frozen embryo imported the Canada and Australia, the same frozen embryo and transport recipient. Recently, last year, we did the same embryo transfer. We find out the 50%. So it's improving the high efficiency. So I already mentioned about again. So you're gonna do increasing some adapted the imported embryo in Indonesia. Firstly, you have to managing the recipient protocol. So it will be good. I don't know what is the recipient in Indonesia, because it's quite different environment, temperature and humidity, and the feeding system is different. So is any maybe some technician or manager visiting our institute? He stayed in over than 2 months. Maybe he will be stayed 2 months in our institute. There he can see how to preparing the recipient in our institute. There will be great chance to improving the embryo recipient program. That'll be the suggestion.
Any other?
Thank you. Okay, thank you, Bubes.
Yeah, okay. While waiting for other participants to have comments on your presentation, maybe I would like to raise also comments, Professor Gudian. According to your research study, are those already in the market? I mean, the cattle that you already mentioned in the previous paper, are they already in the market? And if they are in the market, are they considered to be a GE, genetic engineering livestock? And if they are considered to be a GE animal, do you have any biosafety regulation in order to have them assessed before they are in the market? Thank you.
Yeah, but there will be one of the important issues is commercialization. It's a kind of the food consumption. So in Korea, now it's discussing in government level. So I don't know Indonesia, but recently in Japan and Australia, Brazil, Argentina, Canada, several countries, there will be approved the genome editing pig and cattle and fish and chicken. That will be starting the food consumption. So there will be, I believe that will be genome editing. You have to differentiate the genome editing and conventional genome engineering. Conventional genome engineering means that we have to insert some specific gene. Genome editing, we just deleting the endogenous one.
Yeah.
So this is the genome editing one. Nowadays many countries, there will be divided the non-GMO policy.
Yeah.
That's why the Japan starting and Argentina starting, Brazil starting, the Australia starting, Canada starting.
Yep.
The low level safety issue.
Yeah.
So I maybe in the future means over than probably 5 years later, probably in many countries will be approved the genome editing, the animal including the fish.
Okay. Because in Indonesia, the regulation, the biosafety regulation is only for food crops. there is no regulation for livestock yet.
Yeah.
But according to the technique of the genome editing, gene editing, I think the technique one is considered to be GMO, right?
Yeah,
But the
right.
number three is considered to be considered to be GE, right? Yeah. Okay.
Yeah.
So is there any branding before you go to commercial? To mention that this is the GE1, GE2, or gene editing 1, 2, or 3. Is there any such kind of rules in your regulation?
Currently, I don't have the. I didn't bring the regulation policy, but
Okay. Yeah.
we in government they starting the revise the modification because the dividing the general GMO regulation and genome editing regulation.
Okay. Yeah.
Maybe So maybe you can. I will the next time we're gonna do more discuss about some regulation issue.
Okay. Okay. Colleagues, any other comments or question would you like to raise to Professor Guyang? Let me count 1, 2, 3. Let me see for the next slides. Next. Okay. It seems there is no more response to your lecture, Professor Guyang. At the very beginning of this lecture, we have like 101 people, 81 from the Zoom, and we also have YouTube, running right now, and there are about 20 people are viewers on the YouTube. So it is very interesting of your topics, and we really appreciate your time during your conference in Perth that you still have engaged to our webinar. So I would like to maybe close the session and give opportunity to the M.Si Andalusi to just close the session. Thank you so much.
Okay. Thank you, Mrs. Bess, for leading a very interactive discussion, and for Professor Gujang for a very interesting presentation. So as a token of our appreciation for sharing their valuable knowledge, we will give a certificate for the speaker and moderator. First, to Professor Gujang. So this is the certificate to Professor Gujang as a speaker on the third ROAFERIAN, and for Dr. Insinyur Bess Tisnamurti. Thank you. So, ladies and gentlemen, this is the end of our agenda for today. I'm Andalusi Arini. On behalf of the committee, I would like to say thank you very much to our speaker, moderator, and all participants. See you in the next event. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Bye-bye.
Thank you.
Thank you.
Thank you very much.
We will be in touch with you.
Yes, bye-bye.
Bye.